Hey, is this thing on? All right, what's up? It's Rutherford here. We're going to do a lecture here on test 6. This is the how to make a protein. We call this protein synthesis for G0 cells. Now, G0 cell, like the brain or the muscle cell or any functional cell like your kidneys, your stomach, any cell that's trying to do work inside of your body, it is a fully functional, developed cell. It is differentiated. It is specialized. It has a job to do, like you, and it is going to do work. In order to do work, you must have proteins. Proteins do your work, so you have to be able to make proteins. This is a two-step process. The first step is transcription. The second step is translation. Transcription happens first, translation happens later if you love making proteins. So the first step, transcription, occurs in the nucleus where the DNA in chromatin form is unpacked and ready to be read, aka opened, aka transcribed by a working enzyme, which is a type of protein called RNA polymerase. This RNA polymerase is going to take free mRNA bases that are floating around in the nucleus, and it's going to take them and put together the mRNA strand. This is called transcription, when RNA polymerase builds the mRNA on top of the exposed DNA. So RNA polymerase comes in and opens up the DNA just like a book, untwist, and will build on the exposed DNA. Here we have building on top. It could build on the bottom, but when it's done, it will let go. It has built the mRNA, which will leave the nucleus, and then the DNA will close again. Just like when you're done reading a book, it closes back up. This mRNA, once it leaves the nucleus, we are done with the first step, transcription. Then we move on to our second step of translation, which occurs out in the cytoplasm of the cell. Still inside the cell, but not inside of the nucleus. Once it is in the cytoplasm, the ribosome will come find it. There's two parts of the ribosome. They come and they clamp down on the mRNA, and they're going to read it 3 letters at a time. 3 letters at a time is called a codon. And the ribosome is always going to start with the codon AUG, which codes for the amino acid methionine. Now, these amino acids, there's 20 different amino acids like glutamine and valine, asparagine, aspartic acid, glutamic acid, valine, and tryptophan in turkey. Okay? All of these little amino acids, you either make them or you eat them. And once you have those amino acids digested and absorbed into your stomach, and then it goes to your bloodstream, and then all of your cells have lots of amino acids so they can make the proteins in the way that they want to. And these amino acids in purple here are held by tRNA, which stands for transfer RNA, because it is going to transfer the amino acids to the ribosome. That's their job. And so they are going to float around. The ribosome will bring them in and will take them. They will land on the 3-letter codon, and they will release their amino acid. And the ribosome will take that amino acid and add it on to the growing chain of amino acids called a polypeptide chain. And that chain of amino acids, once the ribosome keeps going 3 letters at a time, eventually it will get to 3 letters that say stop. And then it will be done reading the mRNA. It will be done translating the mRNA. The ribosome will release the chain of amino acids, and we will have a functional working protein. This is the process of protein synthesis. This is the central dogma of biology, where we have DNA, the instructions to make proteins, being opened, read, converted into mRNA, the mRNA then being translated by the ribosome into making a working functional protein. And that's it.